The overall goal of the following experiment is to produce homogeneous levels of human hepatocytes to study human liver biology in vitro. This is initiated by driving human embryonic stem cells, or human ESCs, to definitive endoderm using physiologic stimuli, activin A and Wnt3a. As a second step, definitive endoderm is highly efficiently driven to hepatic endoderm. Next, human ESC-derived hepatic endoderm is matured in vitro to produce functional human hepatocytes. Results from PCR, Western blotting, immunofluorescence, ELISAs, and drug metabolism indicate that this method derives hepatocytes from human ESCs at an efficiency of around 90%.
With this technique, we can in theory generate inexhaustible supplies of primary human hepatocytes, which exhibit appreciable levels of human liver function. In the future, this will benefit accurate prediction of drug toxicity and provide human liver support.
The trick is to drive cellular differentiation at the correct density of human ES cells. Visual demonstration of this method is critical as the correct cell density is essential to generate a homogeneous cell population.
We have trained over 15 individuals over 4 sites internationally to perform this technique successfully.
Begin with a culture of human embryonic stem cells and propagate on matrigel-coated plates with mouse embryonic fibroblast conditioned media, or MEF-CM, supplemented with basic FGF. It is important to carefully characterize the human ESCs maintained prior to hepatic differentiation. For example,